membrane proteins. As we know, there are three types of membrane proteins: peripheral membrane proteins, integral membrane proteins, and anchored membrane proteins. Peripheral membrane proteins are those proteins which are present at the surface of the plasma membrane. It can be toward exoplasmic phase or can toward cytosolic phase. So they are present at the surface of the plasma membrane. Interaction between them is electrostatic interaction and the hydrogen bonding, and they can act as a electron carrier. They play a role in the signal transduction, like in cell signaling, like GPCR, G protein coupled receptor, which is also a peripheral proteins. They act as a enzyme. They act as a cytoskeleton, maintain the shape of the cell. They can anchor other proteins with them. So if we want to study the peripheral proteins, we have to dissociate it with the by treating it with the high salt concentration containing carbonate and in a high pH, so that the interaction between them can be break and the peripheral protein can be removed. Example of peripheral protein: cytochrome C, which is present at the outer surface of the outer side of the inner membrane of the mitochondria. And if we study our basic plasma membrane, we find many peripheral proteins like alpha beta spectrin, actin, band 41, and ankyrin. These are also the peripheral proteins which is present on the RBC plasma membrane. They form an actin meshwork and maintain the RBC shape. And even integral membrane proteins like glycophorin and band 3 is also present in the plasma membrane. So they are integral membrane proteins. Integral membrane proteins are basically embedded within the plasma membrane, and they have a strong hydrophobic interactions because their major proportion of the plasma membrane is the hydrophobic, and they are amphipathic in the nature because the the exoplasmic side and cytosolic side are the polar amino acid, and the hydrophobic core which contain the non-polar amino acid. So it's amphipathic in nature. And the integral membrane proteins has majorly three domains: extracellular domain toward exoplasmic, which has the polar amino acid; transmembrane domain have a non-polar amino acid hydrophobic region that contains; and the cytosolic domain has a polar amino acid. And if we want to study integral membrane proteins, so it's hard to dissociate, so we use ionic detergent and non-ionic detergent. Ionic detergent like SDH, sodium rhodocyl sulfate, and non-ionic detergent Triton X100. SDS denature the protein structure also, while Triton X100 does not denature the protein structure. And if we talk about its types, like integral membrane protein can be of single pass, can be multi-pass. It can be multi-pass also, can be multi-pass. And it can be monotopic and bitopic, polytopic. Single pass which can span the membrane only once, and multi-pass which can span the membrane by multiple times. It can be monotopic, bitopic, and polytopic. Here I have shown transmembrane protein single pass which can pass membrane single times, and it can be type 1 and type 2, and multi-pass which can span the membrane multiple times, type 3 and type 4. Type 2, type 3, and type 4 transmembrane protein. In type 1 transmembrane proteins, amino terminus is towards exoplasmic phase and the carboxyl terminus is toward the cytosolic phase. And while it is opposite in type 2, amino terminus is toward the cytosol and the carboxyl group is toward the exoplasmic phase, but they span the membrane only single time. But in the multipass, in the type 3 and type 4, in type 3 the single polypeptide chain span the membrane multiple times, and in type 4 the several different polypeptide chain span the membrane multiple times. On the basis of, like, if we talk about on the membrane protein topology, it can be monotopic, bitopic, or multitopic. Monotopic is the which span only one monolayer. Okay, it does not impede or does not span the both the monolayer of the plasma membrane. It's only spanning the outer monolayer of the plasma membrane. It in example is cyclooxygenase, COX, which synthesize the prostaglandins and thromboxanes. And biotopic which span the both the leaflet of the plasma membrane. It span outer monolayer and the inner monolayer. It's the biotopic, so it span the both leaflet of the plasma membrane. Example type 1 and type 2. And It's an example like the insulin receptor, glycophorin, LDR receptor. And if we talk about multi-topic, which span the both the monolayer while multiple times they span the plasma membrane. Example GPCR, which span the membrane 7 times, band 3 14 times, and bacterial rhodopsin. So here are the type 3 type 4 include type 3 type 4 transmembrane proteins. Now we talk about the anchored proteins. Lipid anchored proteins means they are not embedded in the plasma membrane, they are attached by some other molecule, like it can be lipid. Okay, they are attached by the lipid which is embedded in the cell membrane, होल्ड्स द प्रोटीन एट द सरफेस, और ये प्रोटीन को सरफेस पे होल्ड करते हैं. जैसे यहाँ पे लिपिड प्रेजेंट है और लिपिड क्या किया हुआ है, इसने प्रोटीन को होल्ड करके रखा हुआ है. सो दे कैन बी होल्ड बाय, लाइक अगर ये फैटी एसिड से अटैच्ड है, सो लॉन्ग चेन्स ऑफ फैटी एसिड कैन बी कॉल्ड है थायोस्टर लिंक्ड fatty acid. Can be palmitic acid or stearic acid. So palmitic is 16 carbon and stearic is 18 carbon. So they by the ester linkage, and the side chain by which they attach is by the cysteine, and it sometimes it can be a serine or thurine. In the example of thioester linked anchored proteins can be GPCR or transferrin protein, and these proteins can be also held by or attached by the some isoprenoid derivatives like farnesyl, which is 15 carbon, and geranyl 20 carbon, and they are called thioether linked prenyl anchored. Example of this is the ras protein, rab protein, and nuclear lamines. So in this the prenyl chain is attached with the cysteine, and there are cysteine residues there, and there is aliphatic amino acid, and then any other amino acid toward the C terminus. Third is the meristymyl anchored amide protein. In this, the meristematic acid, which is of 14 carbon, is attached by amide bond to the alpha amino group of N terminus glycine residue. यहाँ पे glycine residue N terminus के साथ वास के साथ attached रहता है. And the example of this is the G alpha protein and protein kinases. And the fourth one is GPI linked, that is a glyosyl phosphatidyl inositol, and it is also linked by amide linkage, and it is toward the exoplasmic phase. The rest three we have studied like thioester linked fatty acid, and thioether linked prenyl anchored, and the meristatic amide anchored. These are three present at towards the cytosolic side, but GPI linked is towards the exoplasmic side of the plasma membrane. And the lipid anchored protein can also be held with the sterol like cholesterol, and it is also present toward the outer phase of the plasma membrane. Here I have shown the with the diagram. Like if we talk about the thioether linked thioester linked palmitoic or stearic anchored protein fatty acid, it is attached by the cysteine by a cysteine residue, or it can be sometimes serine and thurine. And if there is a attached by the farnesyl or geranyl like isoprenite derivatives, thioether linked prenyl anchor, it is attached by the cysteine residue of a C terminal. And if we by the meristematic acid, and it is attached by the N terminal of the glycine. And GPI linked and cholesterol linked are present toward the exoplasmic phase of the plasma membrane. This is all about the plasma membrane proteins.